All right, let's go over this DNA structure note page real quick. So, first of all, what does DNA stand for? It is deoxyribonucleic acid. Okay. So it is a nucleic acid. It's one of two nucleic acids. Well, it's actually one of multiple nucleic acids found in your body. Two main ones, though, is DNA and RNA, and then we'll also be talking about transfer RNA as well. But this is the main genetic storage molecule in your body. So what is the role of your DNA? In the past, you might say something like it contains your genetic code or codes for proteins, but we're going to get more specific now because we're going to go to a higher level here. So the role of your DNA is to provide the instructions for the primary structure of your proteins. We've talked about in class why it's important that the primary structure is correct on your proteins, and it's not for the primary structure alone, and secondary structure isn't even really that impacted. It's because that tertiary and quaternary structure is highly reliant on the R groups being able to interact properly. And if you have the primary structure incorrect, then the R groups are going to be in the wrong order, and you're not going to get proper folding of those of that protein. So the first thing that it asks, or the next thing it asks you for here, is to sketch and label a deoxyribose molecule. So this is going to be a ring shape. So we can start with that shape here. Okay. Remember that at each intersection, if we're using line structure, that we're assuming there's a carbon there. So there's an oxygen here at the top, then there'd be a carbon here, a carbon here, a carbon here, and a carbon here. Okay. Off of this one here, we will have a hydroxyl group. Here we have a hydrogen. Here we have a hydroxyl group. There's a hydrogen coming off of that one too. And then here, there's a little bit more. So we're actually going to have a carbon. That carbon has a hydroxyl group. It also has another hydrogen on it. And it actually has two. Okay. And that completes our deoxyribose molecule. This is a sugar, and this is the sugar that is found in DNA. All right. So on number 4, I want you to use what we've talked about in class as far as putting monomers together. So we've been doing these dehydration reactions quite a bit this year. And I wanted you to see that it's not just used in putting together sugars or putting together proteins. We see dehydration reactions anytime we start to build a polymer. And DNA is a polymer. It's the biggest polymer in your body. So this is what we're going to be doing here. We're going to try to put a phosphate group together with a deoxyribose sugar. And looking at this now, I just realized that this is actually a ribose sugar. That oxygen should not be there. So my bad on that one. Anyway, so how would we connect this phosphate group to our deoxyribose sugar? Because there's three things that are going to be present in the monomer that makes up DNA. You're going to have a phosphate group, a sugar, and then a nitrogenous base. And that's called a nucleotide, and that's where we're headed towards. All right, so how could we link these together? Well, what we need to do is we need to make water. So how can we make water? If you look here, we have an OH here on the phosphate group. We also have an OH here on this deoxyribose sugar. So we know that we're going to end up linking, we're going to end up making water through these atoms. Now, there's a choice to be made. So we have an OH and another OH. We only need one of those oxygens because it would be H2, and then which one do we pick for our O? This is where you'll want to look at the article and see, okay, how is this thing actually linked? So if you look at the next step, it actually shows you what this step should look like by the time you're done. And you can see that it goes from the oxygen on the phosphate group straight to the carbon. So if we look up here, we need to be going oxygen of the phosphate group straight to the carbon. Well, if you were to make that bond, what is left behind? Oh, I missed. If we were to make that bond, so ignore this first line, what is left behind? Well, you have an oxygen and 2 hydrogens. There's our water molecule. Okay. So then whenever we go to the other side, what is this going to end up looking like? Well, to save time, I'm not going to draw it. I do want you to write it out, but it's going to end up looking exactly like this, where we have water forming, water leaving. So water leaves. Okay. And then that new covalent bond gets formed right here between the carbon and the oxygen. This bond gets a special name. This bond is called a phosphodiester bond. Just like how whenever you link 2 amino acids together, you got a peptide bond. It's just a covalent bond. There's nothing else going on here. But a phosphodiester bond is the covalent bond that links together the phosphate group and the sugar on the DNA molecule. And this is going to end up making the sugar phosphate backbone. Okay, so now we need to put together an entire nucleotide, which is going to be the monomer that makes up nucleic acids. So if we look here, we're going to be doing the same thing. We need to use a dehydration reaction in order to link our sugar phosphate backbone here, connected through the phosphodiester bond, which again, if we were to point to which one that is going to be this bond right here. That's the new one. That is the phosphodiester bond that we just made. Okay. So now we need to link together this adenine, so this would be the A, so this nitrogenous base with this sugar phosphate. All right, so where can we make water? Well, if we look here, we have an OH, so that gets us almost all the way there. We need 1 more hydrogen. Here is a free hydrogen right here. Now, would it make sense? Well, this hydrogen is going to have a positive, it's going to be slightly positively charged because it's connected to a nitrogen. That's going to make it ripe for the picking, and we're going to see right here water be formed. Okay. And that means we're going to get a new bond forming right here between this carbon, where the hydroxyl group left, and the nitrogen, where the hydrogen left. So that's going to be our new bond. So again, I'm not going to redraw this to save time on this video, but what you're going to do is you're going to redraw this whole molecule. The only difference is now the hydroxyl group here and this hydrogen are going to be gone because they just formed water. Water leaves. Okay. So now we're going to have a new bond between this carbon and this nitrogen whenever you redraw it on this side. All right, then we get to number 6. It's asking what are the 3 parts of a nucleotide? Well, nucleotides are the monomer that make up nucleic acids, and their 3 parts are going to be the phosphate group that we just worked with, it also is going to have the deoxyribose sugar, and finally the nitrogenous base. And that makes up our nucleotide, which you just drew in number 5. All right. Label the 4 nitrogenous bases seen in DNA. So what I wanted you to do here was go to the article and figure out which nitrogenous base was which. Now, people typically don't have this memorized. I don't. So this is something that you would look up. What I wanted you to do is realize that there's really kind of 2 options here. There's purines and pyrimidines, and these things have unique structures. So if you were to compare all of these to each other, what we end up seeing is that the guanine is going to be, let's see, checking the article. Here. The guanine has the double bond to oxygen, so that would be this one. Okay, so that would be the G's, the guanine, which means that this has to be the adenine. Okay, and then if we compare these, the thymine has the 2 oxygens. Okay, so guanine, adenine, cytosine, and thymine. So what is the most obvious difference between them? Well, what you see is that the G's and the A's are 2 connected, 2 fused rings, whereas the C's and T's are only made up of 1 ring. And that takes us to number 8. So number 8 says nitrogenous bases are categorized as purines and pyrimidines. Purines have a structure which include 2 fused carbon rings. Okay? Which nitrogenous bases are purines? Well, this is going to be the guanine and the adenine. So you just identified them, and we see that there's 2 fused carbon rings. Which ones are the pyrimidines? This would be C and T, the cytosine and the thymine. Now, I remember back whenever I was in school, I had a teacher that told me this, and it always stuck with me. How do you remember the purines versus the pyrimidines? And he said, just remember that all gods are pure. Okay? So, adenine and guanine are purines. I don't know if it'll help you. It helps me. I'll never forget that. All right. Number 9. Look back at the structure of the nitrogenous bases. Are they polar, and could they experience hydrogen bonds? Well, let's look at these things. Are they polar? We're looking for Knoth-Cloggers, so we're really looking for oxygens and nitrogens connected to hydrogens whenever we're dealing with organic molecules. So nitrogens and carbons, that's definitely polar. Here we have an oxygen and a carbon, that's polar for sure, so guanine's polar. Here on adenine, we have nitrogens and carbons, nitrogens everywhere. Nitrogens are very electronegative compared to carbons and hydrogens, so that's polar. Here we have nitrogens and hydrogens, polar. Double bond to oxygen, very polar. Double bond to oxygen, single bond to oxygen, polar. Nitrogen bonded to hydrogen, polar. So yeah, these things are all polar. Yes, they are polar. Could they experience hydrogen bonding? Well, what is a hydrogen bond? Hydrogen bonds are physical attractions that occur between a partial negative of one molecule and a partial positive of another molecule. If you have 2 polar molecules, yes, they all experience hydrogen bonding. So yes, all nitrogenous bases are polar and can hydrogen bond. Okay, so now we go down to number 10, a process you've heard of before. Complementary base pairing is the phenomenon seen in DNA where purines hydrogen bond with pyrimidines. Use dashed lines to show where hydrogen bonds would form between the 2 pairs of nitrogenous bases below. Then label which nitrogenous bases are which. Okay, so we need to label the nitrogenous bases, and then we also need to show where the hydrogen bonding is going to be happening at. All right. So in order to have hydrogen bonding, we need to know which things are polar and which things are not. So if we look here, we have an oxygen that's connected to carbons, so this thing is going to be partially negative. Here we have a nitrogen connected to a hydrogen, so this thing is going to be partially positive. So what we'll see is a hydrogen bond form between the partial positive on this hydrogen and the partial negative on this oxygen. So here we will have a hydrogen bond. Okay. Then if we look here, we'll have another partial positive here connected to this nitrogen. And due to the hydrogen connected to the nitrogen, so electrons get pulled towards the nitrogen, leaves it partially positive. Here we have a nitrogen connected to carbons, that's going to make it partially negative, and we will get hydrogen bonds forming here. Okay. So we see 2 sets of hydrogen bonds being made, and now we need to label these things as to what they are. This is going to be the adenine You know this because there's no oxygen on it, so it can't be guanine. So these are the A's, so that's adenine. And you would know that adenines, you probably already know this, that A's go to T's. Well, we can verify this, make sure that this is thymine. The two oxygens would indicate that it is, so this is thymine. So this is why A's go to T's. They form hydrogen bonds with each other. Okay. Then if we flip this thing over, we are going to be completing these. Okay, we know now that this has to be either guanine or cysteine, respectively. Well, the guanine is going to be the purine that has the 2 rings, so this has to be guanine, which means that this has to be cysteine. Okay, so where are the hydrogen bonds going to occur? Well, here we have an oxygen connected to carbons, so this thing has to be partially negative here. Here we have hydrogen connected to a nitrogen, so this has to be partially positive. This is why it was so important for us to learn polarity earlier in the year, because we are going to see hydrogen bonding for the rest of the class. I once had a biology teacher tell me, if you don't know the answer to something, just guess hydrogen bonding, because that's why almost everything happens in nature. And he was joking, but you know, there is some truth to that. So we will see a hydrogen bond occurring here. Okay. Then we need to look for more instances where hydrogen bonding could occur. Here we have a nitrogen connected to a hydrogen. That's going to leave this thing partially positive. Here we have a nitrogen connected to nothing but carbons. That'll make this one partially negative, and we can get another hydrogen bond. Remember, these are very weak physical attractions that are easily broken and easily reformed, and they don't require any energy to do so. Okay. Here we have a nitrogen connected to a hydrogen. That's going to be pulling the electrons towards the nitrogen, making the hydrogen partially positive. Here we have an oxygen double bond to carbon. This is going to be very partially negative, and that's going to create another hydrogen bond here. So this is why the G's go to the C's. Okay. 11, looking at these 2 cases of complementary base pairing, which 2 nitrogenous bases are held together through a stronger force? Well, here's the guanine and cytosine interaction. Okay, let's go back and look at the adenine and thymine. What is different? Well, here we only have 2 hydrogen bonds being formed, whereas here we have 3. So that means that the G to C, or the guanine to cytosine interaction, is stronger, and it is due to 3 hydrogen bonds being formed versus just 2. Being formed in A to T interactions. So that means that this is actually a stronger force that is holding together the guanine and the cytosine because of the extra hydrogen bond that the adenines and thymines do not actually contain. Right. 12 is asking, what is 3' and 5' referring to? This is just a way of naming the carbons on really anything that's a carbon compound, but specifically it's used in DNA because it tells you which direction the strands of DNA are running. And we'll talk about this more later, but just to answer this question, what is it referring to? So if we go back to the structure of the deoxyribose sugar, that's where this comes in. Okay, so again we will have oxygen, and then we have this, and here we'll have another carbon, and it has some stuff on it. I'm not going to draw all of the extra things because it doesn't matter for this question. Here we're only referring to carbons. So anytime you name something prime, you're referring to the carbon within the compound. So you always start with whenever you're naming these carbons, you always start your numbering. You're going to pick a landmark, and then you're going to go clockwise from that landmark. And the landmark is always going to be something unique about the structure. So what's unique about this structure? Well, it's this oxygen. So this gives us a landmark to start. So if you start at this oxygen and then start to work clockwise, then we would say that this carbon is 1', this carbon then is 2, this one is 3, this one is 4, and then this one would be 5. Okay. Why does this matter? Well, what we end up seeing is that whenever we're connecting together these nucleotides, they get connected through this sugar. So if you go back and look, whenever we were forming our phosphodiester bond, what were we linking together? Well, we were linking together the hydroxyl group that's on the 5' carbon on the deoxyribose to the phosphate group. Okay. Well, where does this 3' hydroxyl group come in that's on this carbon? Well, that is going to connect to the next phosphate group, which will then connect to the next deoxyribose sugar. So if we were to stack another nucleotide underneath here, we're going to be connecting it to the 3' carbon as we stack these things. And you'll see that this creates an angle. And this is what creates that angular, if you imagine this in 3 dimensions, if we're making this angular movement down, it's going to end up creating that double helix. That's what creates the twist that we see in the structure of the DNA. So what is 5' and 3' referring to? It is referring to the position that certain carbons are at on the deoxyribose sugar. 13, use complementary base pairing to fill in the rest of the DNA molecule, and be sure to label the strand with the correct 5' and 3' orientations. Well, DNA is an anti-parallel molecule, meaning that if it's 5' on this end, then it has to be 3' on the corresponding strand. So this side would then, I'm going to go ahead and turn this the correct orientation. That means this has to be 3' and this has to be 5' because it is anti-parallel. Okay, G's go to C's. We saw Y before. And again, we would have 3 hydrogen bonds occurring here. 1, 2, 3. C's go to G's. T's go to A's. Here we'd only have 2 hydrogen bonds. Okay. G's back to C's. C to G, GC, AT, GC, TA, C, G. All right, so that would be successful complementary base pairing. Okay, so we've gone through a lot more DNA structure now than what you've probably had before. So on 14, it says to describe the structure of human DNA, be thorough, and I already know it's a double helix. You can mention that in your description, but I want more than that. So I'm going to leave this one up to you all. This is the main thing that I'm going to be looking at whenever we collect these and talk about them. I want you to be as thorough as possible, talk about how the backbone is made, so the idea that you have sugars and phosphate groups that are connected together. I want you to be mentioning the idea of complementary base pairing and how we have more hydrogen bonds with the guanines and cytosines than with adenines and thymines, and why we do hydrogen bonds in the first place. I want you to really show an understanding of the structure of the DNA. If you need this to spill over to the back page, then that would be totally fine. All right, I hope this helped. Email me questions if you have them.